So let's look at an example at this. This is, we're going to be talking about enzymes, the lysozyme, which helps to break down peptidoglycan. So peptidoglycan is a polysaccharide. This is a sugar, a string of sugar molecules that are linked together. So you have some sugar monomer that is then linked into a chain. We see these in things like glycogen, for example, which is also another polysaccharide. Peptidoglycan is found in a lot of bacteria cell walls. It's a key component of the bacterial cell walls. And actually, in the beginning of chapter 1, when we talked about differences between bacteria and other eukaryotes, this was one of the things that was mentioned, was this peptidoglycan chain and molecule that is found in the cell walls. So to cleave this peptidoglycan, you can actually lyse the bacteria or kill the bacteria. And an enzyme that many eukaryotes have is called lysozyme, which is essentially an antibacterial enzyme. It's just a enzyme that cleaves peptidoglycan, but by doing this, it lyses bacteria since peptidoglycan is specific to bacteria. So what it's doing here is it is cleaving the bond that holds together two of the sugars within peptidoglycan. You can see we have this one sugar that is highlighted in blue and a second sugar that is highlighted in yellow. And peptidoglycan is just a repeat of these sugars. And lysozyme actually comes in, you can see in the close-up, it's going to cleave this special bond holding together our two sugars using water. So lysozyme and water can cleave this bond. Now again, normally this bond cleavage can occur naturally and spontaneously with just the use of water. But lysozyme is going to catalyze this reaction. It's going to make it happen much faster. And because it's happening much faster, we can quickly potentially lyse bacteria. Whereas if you just had bacteria in water, it's very unlikely. It's not going to happen as spontaneously that their cell wall is just going to spontaneously lyse. Here's a nice just close-up X-ray structure of lysozyme when it is bound to peptidoglycan. So you can see where the chain kind of fits into the notch of the lysozyme. Now, again, this is a close-up. And you can see how it binds into this specific spot between the two sugar groups. How does the cleavage of peptidoglycan by lysozyme happen? It's actually by a series of steps that are Sn2 in nature. So an aspartate, this is just aspartate number 52, on the enzyme of lysozyme can act as a nucleophile. And again, nucleophiles are donating electrons, right? And what that does is it's going to attack the anomeric carbon. This is a carbon attached to that oxygen that holds the molecule together. So this would be an anomeric carbon. The oxygen here that holds the two sugars together, an anomeric carbon is attached to that oxygen. So it bonds onto this anomeric carbon in the first of the steps. Then we have this glutamate 35, number 35, that's going to act as an acid, and it is going to protonate what will be the leaving group in the transition state. So again, this is helping to quicken the reaction by helping with transition state formation. Eventually, in the end, water is going to hydrolyze out the intermediate where we have the sugar kind of bound into the enzyme. leaving us with just our product and essentially regenerating part of the enzyme. In the second step, we're going to see glutamate number 35 again act as a base in this time to deprotonate water in the second step. So I'm going to show you this reaction. And again, this is not a reaction that you need to memorize, but again, try to see where our types of catalysis are, right? See where you can see acid-base catalysis versus your covalent types of catalysis. I remember what Sn2 mechanism is. Sn2 is going to be substitution nucleophilic bimolecular. That's what the 2 is, bimolecular. So two molecules that are involved, two steps involved in this. So you can see again, we have our aspartate that is forming a bond with the carbon. So this is our sugar that is kind of sitting within the enzyme. And we have a covalent intermediate, covalent bond that is formed from the aspartate to bond to this carbon. And then you can see, to help then stabilize this intermediate that is occurring, we also are utilizing the proton on the glutamic acid, the glutamate, number 35 up here. So this is an example of covalent catalysis and an acid-base catalysis that is facilitating this reaction. And again, we're going to focus on here after we remove the half of the sugar through the first part of this stabilization, water is then utilized to essentially regenerate the catalyst and remove the other half of the sugar. So here water comes in, it reprotonates this glutamate, which originally donated its proton. And in doing that, it will then react with the second half of the sugar, helping to break that covalent bond with the aspartate. And in doing that, we've now, again, regenerated our original enzyme state, right? Glutamate is back to its state and aspartate is back to its state. And we have both of our products that have now been formed.